Welcome back to BinaryBio. In our first episode, we explored the atomic revolution of AlphaFold 3. But knowing the shape of a protein is only half the battle. Today, we're zooming out from individual machines to the entire factory flow, the regulatory code of the genome, powered by AlphaGenome. Think of your DNA as a massive library. For decades, we focused on the books, the 2% of our DNA that codes for proteins. But the other 98% is the instruction manual. It contains the switches, the dimmers, and the timers, like enhancers and promoters, that tell your body when to build a heart cell versus a neuron. Because over 98% of genetic variation is non coding, interpreting these instructions is one of the greatest challenges in biology. Until now, AI models faced a fundamental trade off. They were either microscopes, accurate at a single letter level but limited to tiny windows, or satellites, which see a broader context but at a blurry, low resolution. AlphaGenome breaks this trade off. It analyzes 1 million base pairs at once, a context length that captures 99% of all known enhancer gene interactions. The architecture of AlphaGenome is designed to act like a powerful translator that reads 1 million characters of DNA code and identifies where specific biological activities occur. It uses a UNet inspired design, which means the data is squeezed down to find patterns and then expanded back up to provide detailed results. Here is a breakdown of the four main parts in simple terms. The sequence encoder. The encoder takes this 1 megabase sequence and gradually zooms out. As it moves through different layers, it condenses the information. It turns every 128 letters of DNA into a single mathematical summary, an embedding. The transformer tower. DNA doesn't work in isolation. A piece of code in one spot might control something very far away. The transformer tower consists of stacked blocks that use attention mechanisms. This allows the model to look at a specific gene and simultaneously pay attention to a control switch, an enhancer, located hundreds of thousands of base pairs away. Pairwise interaction blocks. DNA isn't a flat line, it folds into 3D shapes. These blocks work alongside the transformers to handle spatial data like high C contact maps. They help the model understand which parts of the DNA are physically touching each other in 3D space, which is often how genes are turned on or off. The sequence decoder. Once the model understands the context, it needs to provide answers at a very specific level, sometimes down to a single base pair. The decoder takes the summarized data and zooms back in. Skip connections. To make sure it doesn't lose the fine details it started with, it uses shortcuts that bring high resolution information directly from the encoder to the decoder. Finally, the model uses specialized heads, like different apps on a phone, to output specific predictions, such as where proteins will bind or how much of a certain gene will be produced. Processing a million bases is a massive memory challenge, so AlphaGenome uses sequence parallelism, splitting the workload across 8 interconnected TPU chips. To make it fast enough for daily research, the team used knowledge distillation. They trained a single student model to mimic 64 specialized teacher models. Let's see this in action with a case of T cell acute lymphoblastic leukemia. Researchers used AlphaGenome to virtually screen the TL1 oncogene. The model accurately predicted how a tiny insertion, thousands of bases away, acts like a hot wire on a car, flipping a cancer driving gene to permanently on. AlphaGenome is a bridge between the digital world of AI and the biological reality of our cells. With an open API for researchers, we are moving from a world where we guess what a mutation does to a world where we can simulate the consequences of every single letter in the human genome. If you found this video helpful, hit the like button and subscribe to BinaryBio. We're here to help you navigate the intersection of code and biology. Thanks for watching, and I'll see you in the next one.